So once again, this is the plot that was initially shown about the tumor hallmarks, and these are common phenotypic traits of cancers that emerge following Darwinian selection and confer a selective growth advantage. You'll notice that most of these, evading apoptosis, self-sufficiency and growth signals, insensitivity to anti-growth signals, and limitless replicative potential, have to do with that balance between cell death and proliferation that allows cells to then clonally expand and become a tumor. A couple of these other ones have to do with later adaptations for tissue invasion and metastasis and sustained angiogenesis. And as was noted earlier, more recently, there's been a discussion of additional emerging hallmarks, which include deregulation of cellular energetics, the glycolytic phenotype, and the genomic instability and mutator phenotype. And there is a relationship between these two because hyperglycolytic activity leads to tumor acidosis, which is a known cause of mutation and classigenesis, genomic instability. A couple of the other ones, avoiding immune destruction and tumor-promoting inflammation, are interesting and are potential new directions that we can go in molecular imaging. So functional equivalence, a second evolutionary principle is that the environment selects for phenotype, not genotype. And multiple genetic backgrounds can lead to similar phenotypes. So thus, cancer can be genetically heterogeneous, yet have common phenotypes caused by convergent evolution. And so what's important about this is that rather than necessarily focusing on the genes, it may be important for us in terms of cancer imaging and therapy to focus on these phenotypes and find ways to actually treat or image these phenotypes as opposed to specific genetic markers or pathways. A while back, Gillies and Gatenby presented the hallmarks, the hallmarks in a slightly different way in terms of overcoming adaptive barriers to carcinogenesis. And you can see the hallmarks were listed across here. Although this was published before the second update of the hallmarks, they were already talking about this energetics hallmark as well. And so in terms of these hallmarks related to the pathway of carcinogenesis, they observed a number of potential barriers that tumors need to adapt to as they progress through carcinogenesis. And for example, apoptosis with loss of basement membrane contract needs to be overcome in order to move on from normal tissue to hyperplastic tissue, which is developing a resistance to contact inhibition and anoecus. A next barrier had to do with inadequate growth promotion, and the way the phenotypic strategy for overcoming that barrier was upregulation of growth factors or receptors, leading to this hallmark phenotype. And so you can see along the way there's a series of these barriers and a series of phenotypic strategies that where the first set here all have to do with that balance between proliferation and cell death leading to clonal expansion. And the later ones have to do with invasion and metastasis. Okay, so how do you image this balance between growth and cell death, the proliferative advantage? there have been a few ways that people have gone about this. Once again, I'm talking about this early part of those barriers that were described in the last slide. So after the fact, ex vivo histology can be used to look for cell death morphologies and cell death markers. MRI and ultrasound can be used to follow the growth of tumors, so the tumor growth rate can be followed in vivo. And diffusion-weighted MRI can be used to as a measure of cellularity and this either loss of cell death or can follow cell death in the context of therapy. Also, there are metabolic markers for proliferation, such as choline, that can be followed by magnetic resonance spectroscopy. So here's a study we did a while back where we were looking in animal xenografts in vivo, diffusion-weighted MRI. These are breast tumors, breast cancer tumor lines that were xenografted into skid mice. And you can see that in the untreated tumors, the apparent diffusion coefficient map, which is a measure of the level of diffusion, these values in this outer periphery of very dense cells was really quite low. And so this is an example of this hallmark where the cell death rate is quite low. This is a measure of cellularity. And following treatment, you could see cell death markers, and in the periphery, the diffusion shifted a little bit higher. And this change was proportional to the dose and was significant. Here's a recent study in the brain of a glioblastoma multiforme that's highly proliferative. And you can see by MRI and FLAIR, you can see where the tumor is. But when you look at diffusion-weighted MRI, you can see that in the area of the tumor, it's quite bright. And in the case of diffusion, that means diffusion is very low. And with the apparent diffusion coefficient map, it's actually quite dark in the area of the tumor, and this also indicates that diffusion is quite low. And so this is an example of what we're talking about with this highly proliferative tumor with very low diffusion rates. So choline has been is a marker for proliferation, and it's been used to study the level of proliferation in tumors. This is a recent study by Jagannathan's group, where he looked at total choline. It's a single voxel magnetic resonance spectroscopy study. And you can see this is the total choline peak. This is the voxel area and in a malignant breast tumor. And this total choline peak includes glycerophosphocholone. It's a composite peak. It includes glycerophosphocholone, phosphocholine, and choline. And you can see that it's highly elevated in this proliferative malignant tumor, whereas in the benign tumor, it's much lower, and in normal tissue, it's much lower. And in this study, these differences in patients were significant from the malignant to the benign and the malignant to the normal tissues. The same group has also done magnetic resonance spectroscopic imaging, and just to highlight what they've done here, here's a spectral map of these voxels in the tumor, and if you see and here's the tumor and here's the normal surrounding tissue, and if you see the spectra that are in each voxel, the choline peak is quite high within the tumor. And as it moves outwards towards this voxel where there are partial volumes, you see that the choline peak decreases and that these other peaks begin to increase that are associated with normal non-proliferating tissue. And by the time the voxel has completely left the tumor, the choline peak is almost non-detectable. And from those data, they were able to produce a map, a metabolic map of choline in this tumor. So this unchecked growth then can lead to chronic hypoxia and upregulated glycolysis. So, let's see, what am I doing? If you look at these breast tumors, these early tumors are all encapsulated in a basement membrane, and they're avascular. And you can see the vessels surrounding these small tumors. And what we observe is that the tumor grows inward so that the diffusion of oxygen limit is reached. And then there is a hypoxic region within the tumor. We can assume possibly that these cells that are still surviving right at the edge are very hypoxic, and they are undergoing selection for adaptation to that environment. So what are ways that people have gone about imaging tumor hypoxia and direct imaging of oxygen? So F-meso has been used for PET imaging, and this agent is deposited in regions of hypoxia. So it builds up in areas of hypoxia and then can be detected by PET imaging. But until recently, there hasn't been a way to actually directly measure oxygen in tumors other than by inserting a probe very invasively. But now a group is doing oxygen EPRI, electron paramagnetic resonance imaging, as a direct non-invasive measure of oxygen in vivo. And other groups are looking at biological markers of hypoxia, such as CA9, and developing molecular imaging probes that are targeting these markers. So the Cherukuri group at the NCI is doing this EPR oxygen method, and this is done by using a paramagnetic tracer where in the presence of oxygen, the line width of the tracer broadens proportionally with the level of oxygen. So the line width generated by the tracer, as it broadens, it does this in a proportional way with oxygen levels. And so very nice images are acquired sensing oxygen in real time. Oops, wrong one. And as you can see, there's very nice images showing the heterogeneity and different levels of oxygen within these tumors. And this can be co-registered with the morphological MRI images, and it can be done over time and in 3 dimensions with pretty good spatial resolution. So imaging carbonic anhydrase 9. Carbonic anhydrase 9 is under the control of the hypoxia-inducible factor. Nargis Tafrishi, this image is taken from her poster, and she's going to be presenting this whole study later today in the poster session. But she made a probe, a monoclonal antibody probe that was specific for carbonic anhydrase 9, conjugated it to a fluorochrome, and then took animals, grew tumors, and incubated them or allowed them to grow in conditions of hypoxia, normoxia, and hyperoxia. And the subsequent uptake of this probe following growth in these conditions showed a significant increase in hypoxia and a decrease in hyperoxia. And this was correlated with pimenidazole, which is builds up inside tumors as well. And so the same relationship of significant increases in hypoxia and decreases in normoxia was observed. there's this process of adaptation to these hypoxic regions and the resulting acidosis. And here you can see, once again, these early encapsulated tumors that near the necrotic edge in this area that is likely hypoxic. Carbonic anhydrase 9 is being expressed. And also the glucose transporter, which is also under control of the HIF promoter, is also expressed in these regions. And this is called the Pasteur effect, where in hypoxic regions, glycolysis is activated. That's something that happens in normal cells. But what doesn't happen in normal cells is an example down here where you can see that this glucose transporter it continues to be expressed in cells that then invade outside back into the oxygenic area, and yet the glucose transporter stays elevated. And this is an example of the Warburg effect that's observed in cancer cells, where cancer cells have a glycolytic phenotype. They have this propensity to continue with glycolysis even in the presence of oxygen. Here's another example of this, where one patient, by using FDG PET to measure glucose uptake or FMESO PET to measure hypoxia, you can see that this patient, as you would predict, has a hypoxic tumor with high glucose uptake. But here's another patient that has very low hypoxia and yet high uptake. So this is another example of the Warburg effect. So this is a common observation that glycolysis is a common trait among almost all metastases. Almost all metastases are glucose avid and can be observed by FDG PET. So Gillies and Gatenby, a while back, presented a model about how this adaptation could occur. As you can see, cells begin to grow, and they grow out to their oxygen diffusion limit, and they stop there for a while, but then they're in this hypoxic region, and they're undergoing selection in that area, and they're able then to become glycolytic and grow beyond that. But then they reach what they call an acidosis limit. So these cells are unable to grow in these acidic conditions. But once again, they're under selection for not only low oxygen, but also growth and low pH. And at some point, they become able to continue growing even in these acidic conditions. And you see that these are the cells that then are able to outgrow. And there's a lot of evidence now that shows that low pH from the tumor, from these cells, is what allows the basement membrane to be degraded and allows invasion beyond the basement membrane and metastasis. So how do we go about imaging tumor pH? There are magnetic resonance spectroscopy methods and acidoCEST, chemical exchange saturation transfer MRI, and there are optical intravital imaging methods that are being used, among others. So here's an example of a study that was done in the Gillies group a while back. And it's using phosphorus magnetic resonance spectroscopy, single voxel. You can see here's a tumor, a xenograft tumor. Here's the voxel. An agent, 3-APP, is used, and its chemical shift is pH dependent. And it is excluded to the extracellular environment. The inorganic phosphate peak is a measure of the intracellular pH, and its chemical shift is also pH dependent. And so by determining this spectrum and quantifying these peaks, they're able to determine the pH in the extracellular and intracellular environments. And they did this in a number of tumor xenografts and could see that in almost all of them, the pH, it was lower, much lower in the extracellular environment relative to the intracellular. The same group also worked with a group in Spain who developed IEPA, which is a pH-dependent indicator for a proton MRSI pH mapping experiment. And here's the result of that experiment, where they were able to show heterogeneity of tumor pH throughout the tumor with a range from 6.8 to 6.4. Okay, acidoCEST is being done in the Pagel group at the University of Arizona, and it's done using a contrast agent, iopramide, which has an amine and an amide group, where during saturation transfer there's a pH-dependent saturation of the water signal. And you can see that in the case of, so this used to have pH symbols across the side here, but in the case of, I believe, lower pH, this ratio is in one direction, and then as the pH increases, the ratio becomes the other direction. These peaks can then, or valleys, can then be quantified by line fitting, and the ratio then determines, this is missing a lot of detail in the slide, but there is a plot here that has, sorry, what? Oh, sorry. I apologize. Yeah, so there's a lot of detail missing on this plot. I don't know what happened. Evidently, the system dropped some of the lines, but there's a line fit here with a very nice R-squared value, and it shows basically that with acidoCEST, the precision is really approaching that of a pH meter, and this is done in vivo. And so here's a study done by the Pagel group where they imaged animals using acidoCEST. These are breast tumor xenograft models. You can see the tumors, and here's the bladder. And one group of animals was given water, just regular drinking water, and another group of animals was given water with bicarbonate, which buffers the pH throughout the animal, including the tumor and the bladder. And you can see the acidoCEST was able to measure the pH in the bladder, and this is before bicarb, showing a much lower pH, and then the bicarbonate then buffered the pH in the bladder in both of these animals. And in the animals whose tumors had low pH, the bicarb was able to buffer the pH, raise it to neutral levels, and this isn't so much observed in animals that didn't already have lower pH, but that's to be expected because the buffer doesn't raise the pH per se, it just buffers it to neutral pH. A third way to look at pH imaging in vivo involves looking at dorsal window chamber experiments where a window chamber is placed on the back of a mouse, and under the cover slip a tumor is implanted so that this this tumor can then be imaged using a confocal microscope. And the result of this study demonstrated that by looking at how the tumor grew, you can see this is how the tumor started out, and then after another week, the tumor grew out in this direction. By using a pH-dependent fluorescent dye, SNARF, they're able to measure the pH gradient as well, and they're able to show that in the regions of low pH, this is the direction that the tumor grew into the surrounding tissue. And you can see this is plotted here where at low pH, the tumor growth was the highest, and at high pH or neutral pH, the tumor growth was the lowest. So this leads to the acid-mediated invasion hypothesis, where tumors that secrete acid can invade, and that this acid is glucose-derived. And here is an image of a metastasis with the surrounding tissue. And in this region, it's been shown that the extracellular matrix has been degraded, and that this is the region of high pH. And there have been a number of other studies showing that pH has a strong relationship to invasion. And you can see here the proposed mechanism where the tumor secretes, lowers the surrounding pH, and then that prepares the environment, and then the cells can invade. So angiogenesis, the last block to be overcome, can be imaged by histology, looking at perfusion and vascular markers, but also by DCE-MRI, looking at vascular permeability and uptake. Here's a study done by Reddick where osteosarcomas, DCE-MRI was performed on osteosarcomas showing heterogeneous perfusion. So here's a plot of region A, you can see right here, that has low permeability, low volume, and B that has high permeability, low volume, vascular volume, and D with high permeability and high volume, vascular volume, and C, low permeability and high vascular volume. So this demonstrates the heterogeneity of vascular permeability throughout the tumor and heterogeneity in the types of environments that are encountered by tumor cells. Here's another study by Benedict Jordan who looked at xenografts in mice, and you can see that the control tumor had very high vascular permeability, whereas following treatment with PX478 that inhibits HIF1 alpha, this vascular permeability was greatly decreased. So I guess I'm just going to wrap it up and conclude here, briefly talking about some of these other hallmarks. I think we covered the disrupted glycolytic phenotype quite well and its relationship to genomic instability. But although there's been quite a bit of work in terms of tracking immune cells with MRI and other methods, I think that this avoiding immune destruction and tumor promoting inflammation are two areas where we can look for new ways to image this by looking for tumor markers or ways to actually identify these regions related to others. So that's, I'd like to conclude the talk, and thank you for coming and participating.